Laura Roselli here, March 2021. Today I want to talk about variants, specifically variants of coronavirus 2. And I first want to back up a moment so that we can talk about what viruses are and how variants can be created. Viruses are parasitic. That means that a virus needs a host in order to make more copies of itself. A virus is a very simple structure. We're not considering them living. They are made up of a protein coat, and these little proteins on the outside are called part of the protein coat or capsid. Specifically, we're going to talk about this spike protein on the outside. Your immune system actually only really sees the outside of this virus. So that's typically what we're trying to train your immune system in, is recognizing these proteins. Also on the inside of the virus, you have some genetic material. And in the case of SARS coronavirus 2, it's RNA, but it can be DNA as well. So this genetic material and some proteins on the outside are basically it. That's really all that viruses are. In order to make copies of themselves, as I mentioned, they need to get into a host. And the host is one of our cells, is us or one of our cells. And so this here might be a human cell that is the host for a particular viruses. And viruses have specific cells that they want to get into. Now, SARS coronavirus 2 has a lot of different cells that it wants to get into. And specifically, it's trying to get into this ACE2 protein. So there's a protein on my cell, and then there is a protein on the virus. And those two have to connect in some way in order for the virus and its genetic material to be allowed into the host cell. Once inside that host cell, this cell gets taken over. The viral machinery or the cellular machinery starts reading this viral recipe, this viral genetic material. And it's like a factory. And the cell is the factory, but the virus has hijacked the factory and convinced it that it should make a whole bunch of these coronaviruses instead of doing what our cells normally do. Now, let's talk about the variants a little bit. So I want you to imagine you have one virus getting inside your host and many, many viruses coming out. It's not like you have 1 to 1 or 1 to 10. Many viruses leave the host. And each time that those viruses are made, there's a potential that there's a slight change in the virus and in the proteins. So again, let's revisit the virus and these proteins here, the spike proteins. What can happen is when the cell starts to make copies of this, there's some changes, some mistakes in the RNA as that RNA is copied. And what that may do is that may create a protein on the outside that is slightly different than the one that infected the host. So when you talk about variants, we're talking about mutated forms. The proteins that are made by the recipes of RNA or DNA, the DNA or RNA is changed. So the proteins that are made are now different. Those variants may or may not be recognized by your immune system, depending on what your immune system has seen naturally or artificially in the case of the vaccines that we have. So there are a few variants that are already being discussed, and we have looked at J&J's vaccine and whether they can fight off these other variants. And the key is that the less we allow the virus to infect hosts like us, the less chance that these variants are made. So the quicker we can get to a level of immunity in our population, that the less likely the new variants will come to be. Hopefully that makes sense to you, and we'll talk more about the different variants and the vaccines that may fight those variants in a future lecture.